Cell-mediated immunity is one way the body fights an infection or cancer. Two types of cells are involved in cell-mediated immunity. Macrophages, which engulf and destroy pathogens, and T cells. There are three types of T cells: helper T cells, killer T cells, and suppressor T cells. Pathogens are disease-causing organisms. Pathogens have unique molecules on their cell surfaces called antigens. These body cells are about to be infected by a pathogen. The pathogen enters the cell where it disassembles. Antigens appear on the surface of the infected cell. Some of the pathogens are attacked by the body's macrophages before they can invade a cell. Once a pathogen is destroyed, pieces of the antigens are displayed on the surface of the macrophage. The rest of the pathogen is destroyed. A site on the surface of a helper T cell recognizes the antigen on the macrophage. Once the helper T cell binds to the macrophage at its recognition site, the macrophage sends a chemical signal to the helper T cell. The helper T cell is stimulated to divide many times, producing clones of identical helper T cells. Each helper T cell has the antigen recognition site of its parent. The helper T cells secrete a chemical substance that activates killer T cells. The activated killer T cells recognize the antigen on the surface of the infected cell and bind to it. The killer T cell releases a substance called perforin, which makes holes in the membrane of the infected cell. The cytoplasm of the cell erupts through these openings and the cell is destroyed. The killer T cell moves on to another cell, and the process of recognizing the antigen, binding, and destroying the cell is repeated. When antigens are no longer present, the suppressor T cells turn off the immune response by releasing a chemical that suppresses the activity of the other T cells.